Hello everyone, welcome back to my channel. Thank you for tuning in for another exciting video today. So, in context of RNA sequencing analysis, DESeq2 has emerged as a powerful tool for identifying differentially expressed genes. And previously, I have created a couple of videos on my channel talking about how DESeq2 works, and I've also demonstrated how one can perform differential expression analysis using DESeq2. If you haven't watched those previous videos, I will recommend you to check them out. I will be adding the link to those videos in the description section below. One of the key strengths of DESeq2 is its ability to handle complex experimental designs, which often involves multiple factors that may interact with each other. So in this video, we'll break down the concept of interaction terms in DESeq2 and explain and understand how to include them in our analysis and demonstrate how to interpret the results. By the end of this video, we will be having a solid grasp on how to leverage the interaction terms to enhance our RNA sequencing analysis. So, familiarizing ourselves with the ideas like main effect and interaction term while setting up the design of our DESeq2 experiment or analysis. So, here we have two factors. We have genotype and treatment. Within each of these factors, we have two groups. We have wild type and mutant mice in genotype, and we have control and treated mice in the treatment. So the main effect represents the overall effect of a single factor on gene expression averaged across all levels of other factors in the experiment. So here we are looking at just one factor at a time. Similarly, we can also look at the treatment, that is the other factor in the experiment, where we are comparing the treated versus the control mice and essentially trying to identify the differentially expressed genes between these groups. So together we are trying to estimate the effect of each factor independent of the other factor. However, interaction terms capture the effect of one factor on gene expression depending on the level of another factor. So in this case, we are trying to understand whether the treatment effect is dependent on the genotype. So interaction terms are usually written as the name of one factor colon the other factor. So this is how we indicate that we are interested in studying the interaction between these factors. So this is just to give you a brief overall idea on different ways the design can be set up and different questions that can help you answer and understand the effect of these factors. Moving on from the following slide, we will go case by case, trying to understand a very simple experimental setup with just one factor and two groups, and going into more complex designs and understanding what kind of questions those kind of designs can help us answer. So let's consider the first case, which is a very simple experimental design, which contains just one factor, that is genotype, with two groups, that is wild type and the mutant mice. And let's say the goal of the experiment is to identify genes that are differentially expressed between mutant and the wild type mice. So the design here is going to be very simple. We are just going to provide the genotype because we are interested in comparing the groups amongst that factor. Here is the table portraying or labeling the mice or the samples which are wild type and the ones that are mutant. And here is the design matrix, which essentially encodes the experimental conditions. So the design matrix is used to represent the different experimental conditions or factors that one want to analyze. So here we have samples from one factor but different conditions, that is mutant and genotype. So all the samples of the mice that are mutant are labeled or encoded as 1. The rest of them are encoded as 0. And this design matrix is a part of the linear model that DESeq2 fits to the count data. So each row here in the matrix corresponds to the sample, and the columns correspond to the coefficient, that is intercept or the condition or the genotype in this case. So when we run DESeq2 for this experiment, what we get is essentially a resulting output of differentially expressed genes in mutant versus the wild type mice. And this is what essentially the results look like. We have genes here which are differentially expressed with the log fold changes and other relevant statistics in the mutant mice versus the wild type. We have gone over the stats in detail in one of my previous DESeq2 videos, so I'm not going to go into the details of that. I will be adding those links in the description section. So if you're interested in learning more about how to conduct a DESeq2 analysis, I will recommend you to check those videos out. So adding in a little bit more complexity, let's say we want to compare across multiple groups in one factor. So from the previous case, again, here we have one factor, but we have more than two groups. So we have three genotype here. And let's say if the goal of the analysis is to identify genes that are differentially expressed in genotype 2 and genotype 3 compared to genotype 1, considering genotype 1 as the reference or the baseline. We can still continue to use the same design by providing that factor to the design. So here we have the call data, the metadata table labeling the samples belonging to the different genotypes. And here is the design matrix encoding each sample based on the different genotype. And on running DESeq2, we essentially end up with differentially expressed tables comparing group 2 versus 1 and 3 versus 1. So this essentially achieves what we were hoping to identify or get from this experiment. So if there is comparison between multiple groups in one factor, then we would still essentially provide just that factor to the design to perform comparisons across the group. So for the next case, let's say we have two factors, genotype and treatment, each with two groups. So this is something that we have looked at previously. And let's say the goal of the analysis is to study the effect of each factor independently. So we want to study which genes are differentially expressed between wild type and mutant genotype. And we also want to study which genes are differentially expressed between treatment and control mice. So when we want to study the effect of the factors independently, we set up the design something like this, where we provide one factor plus the other factor. So here we have the metadata table that shows what values or what each sample belongs to for each of the factor in each of the factor. And this is a design matrix with the relevant encodings. So in the first column is the intercept, the second column is the genotype factor, and the third column is the treatment factor. So for the genotype factor, all the samples that are of mutant genotype are encoded as 1, the wild type are encoded as 0. And for the treatment factor, all the samples that have undergone treatment are encoded as 1, and the control samples are encoded as 0. So when DESeq2 is run on this data, essentially what we end up is with two different tables showing differentially or providing differentially expressed genes in one factor, that is mutant versus wild type, and in another factor, that is condition, that is treatment versus control. So if the goal of the analysis is to compare or understand the effect of each factor independently, then you would want to set up your design experiment something like this. Now it is important to note that there is a minor difference when we set up our design as genotype plus treatment versus treatment plus genotype. So when we set up our design as genotype plus treatment, the main effect here is treatment while controlling for genotype. What this means is that the model will adjust for the genotype first and then assess the treatment effect. So you will interpret the treatment effect as the difference in expression between treatment and control, having already accounted for any difference between mutant and wild type. Similarly, for the design where we have treatment plus genotype, the main effect is genotype controlling for treatment, meaning the model adjusts for treatment first and then assesses the genotype effect. So you will interpret the genotype effect as the difference in expression between mutant and wild type genotype, having already accounted for any differences between treatment and control. A quick note here is that this order will not change the fit of the model, will not change the statistic. It only matters in terms of the interpretability. Both these models will give you overall the same statistical conclusions, but the order in which you interpret the effect will differ based on the formula that is used. So this distinction is important for correctly conveying your biological hypothesis and understanding the relationship between the factors. So before adding more complexity, let us understand what are interaction terms. So this experiment here involves three genotypes, genotype 1, 2 and 3, and two conditions, condition A and B. For a particular gene, gene 1, the experiment records the expression levels on the y-axis as log 2 counts plus 1, which are measured across all the genotypes for both the conditions. The black dots here represent individual samples, and the red lines connect the average expression levels for conditions A and B across each genotype. Let's say this is our design, and this is the term that is the interaction term. So let us understand this in a little more detail. So this interaction term essentially answers the question, is the condition effect different across genotype? Does the condition effect vary across the genotype? So the condition effect for any genotype is essentially a sum of the main effect and the interaction term. The main effect here being the how much gene expression changes on average when you go from condition A to condition B, ignoring genotype. And for the interaction term, let's take an example to understand the condition effect for genotype 2. So basically the here we are interested in understanding whether the condition effect is different in genotype 2. So here the interaction term would be genotype 2 condition B, which essentially tells us how much more different or how much more or less the effect of condition B is in genotype 2 compared to genotype 1, considering genotype 1 as the reference. This is essentially what will give us the condition effect for genotype 2 when we run the DESeq with this design experiment. So now that we understand what are interaction terms and what questions do they help us to answer, let us understand when to use these interaction terms. So continuing with the same example that we spoke about in the previous slide, here we observe that each genotype starts with a different baseline level of gene expression under condition A. So the genotype, the baseline expression under condition A is the lowest in genotype 1. It's slightly highest in genotype 2 and highest in genotype 3. The effect of going from condition A to B is consistent across all the genotypes. The increase in gene expression is about the same, is close to 2 log-fold change for each genotype. So let's say if we model this data without interaction terms, that is using only genotype and condition without the interaction term, we are assuming the effect of condition is same across all the genotype, which seems appropriate in this case since the condition effect is consistent. But let's say if we were to use the interaction term, that is, we use genotype plus condition with the interaction term, we are modeling the possibility that the effect of condition could be different for each genotype. However, in this case, since the effect of condition A to B is about the same for all the genotype, the interaction terms for genotype 2 and 3 will be close to 0 or 0, indicating that there is no significant difference in condition effect across the genotype. So let us do the math and see and calculate the interaction terms for 2 and 3 to see if that holds true. So in this case, the main effect of condition B would be the average expression at B minus the average expression at A. That is 10 minus 8 is going to be 2 at the reference level. So this is the main effect condition. Now let's calculate the interaction term for each genotype. The interaction term for genotype 1 is going to be 0 because it's the reference and there is no additional interaction effect. For the genotype 2, the condition effect for genotype 2 again would be the difference of the average expression at condition B minus the average expression at condition A, which is going to be 11 minus 9, which is equivalent to 2. Now, the condition effect for 2 is equivalent to the main effect, meaning that there is no interaction and hence the interaction term is 0. Similarly, for genotype 3, the condition effect for 3 is going to be the expression, the average expression of B minus the average expression at A. Again, this is 12 minus 10, that is 2, equivalent to the overall main effect condition, indicating that there is no interaction, and the interaction term for this is also 0. Since the effect of condition A to B is same across all the genotypes, we can model this data without the use of interaction terms. Now let's consider another case. Here, each genotype again starts with a different baseline level of gene expression at condition A. This part is similar to the first case. However, the main difference here is that the effect of moving from condition A to condition B, which varies across the genotypes. For genotype 1, there is an increase from condition A, increase in expression for condition going from condition A to condition B. For genotype 2, again we see the increase, but the increase is larger compared to 1. However, for genotype 3, the expression actually decreases as the line is going down, when we are going from condition A to condition B. So if we were to model this data without interaction terms, just using genotype plus condition, we assume that the effect of condition is same across all the genotype, which is not true in this case. So adding interaction term in this case, that is genotype plus condition plus the interaction terms, allow us to model the different effects of condition across genotypes. So the model first accounts for the overall effect of condition for the reference genotype, that is genotype 1, which is log 2 fold change of 2. The interaction term then adjusts this effect for other genotypes. So for genotype 2 has an additional increase of 1, making the total change of 3, and the genotype 3 has a decrease of 4, making the total change of 2. Let me show you the similar math behind this. So let us also calculate interaction terms for this case. So here the main effect for the condition B is going to be the average expression of at B minus the average expression of at A. So this is the main effect condition is going to be 2. Now calculating the interaction term is going to be again 0 for the reference because there is no additional interaction. However, the condition effect for 2 is going to be the expression at B minus the expression at A, which is going to be 12 minus 9, which is going to be 3. And the interaction term is going to be the condition effect, that is 3, minus the main effect, that is 2. So the interaction term here is going to be 1. For genotype 3, the condition effect for 3 is again going to be expression at B minus the expression at A, which is going to be negative 2. And the interaction term here is going to be the condition effect for 3, that is negative 2 minus 2, which gives us the interaction term as minus 4. So in this example, the condition effect varies across genotypes. So including the interaction term in the model is important. These terms help to capture the differences in how each genotype responds to the condition change. Without them, we would miss out on understanding these different responses. So now taking a look at another case where we have two factors but with multiple groups. So let's say we have genotype and treatment, and genotype has three groups, genotype 1, 2 and 3. And under the treatment, we have two groups, control and treatment. And if the goal is to understand the effect of condition for different sets of samples or to extract the condition effect specifically for each genotype, the authors of DESeq have recommended a simpler approach that we can simply concatenate both these factors into another factor and use that new factor for the design. So we could model the data. In this case, we would not need to use interaction terms because we are simply interested in the overall effect of each factor and not necessarily how the effect might interact or change depending on each other. Only then this approach would make sense. So again, we create another group and provide that to the design, and this is the corresponding design matrix. And once we run DESeq, we get access to different comparisons across different set of groups and samples that we can make. And again, I want to reiterate that this is an approach if we are just interested in understanding the overall effect of each factor, but not necessarily how they might interact or change depending on each other. So just because we have multiple factors and multiple groups might not warrant the need to use interaction terms. The interaction terms and the design setup is highly dependent on the question that you are asking. Considering another scenario with the same experimental setup, in this case, let's say the question that we are asking is that we want to understand the effect of treatment or condition depending on the genotype, or whether we want to understand the treatment varies with different genotypes or changes across the genotype. In that case, when we want to study the effect of one factor depending on the level of another factor, we must use interaction terms. So in this case, let's say if we set up the design, genotype plus treatment and the interaction term, we will essentially be using both the factors, and this is the design matrix. And once we run DESeq, what we get is the overall effect of genotype, treatment, and the interaction terms. Again, the interaction terms would be the condition effect or the treatment effect for that particular genotype plus the overall effect. Also wanted to quickly point out the distinction between just using or providing the interaction term to the design and providing the main effects plus the interaction terms. So in this case, the model accounts only for the interaction effects between the factors. In this case, the model effects accounts for the main effect along with the interaction between the factors. This should be used only when we are interested in the interaction between genotype and treatment and do not care about the individual effects of genotype and treatment. An example would be if the primary hypothesis is that the effect of a treatment is different for each genotype, but we are not specifically interested in the individual effects of genotype and treatment alone. For this case, this could be used when we are interested in understanding both the individual effects of genotype and treatment as well as their combined effect. So if the hypothesis involves understanding how each genotype affects the gene expression, how each treatment affects the gene expression, and whether the effect of the treatment depends on the genotype. So in this case, the model will only consider the specific combination and provide estimates of each combination without separating out the individual effects of genotype and treatment. And in this case, the model will separately estimate the baseline expression level, that is intercept, the main effect of genotype, the main effect of treatment, and the interaction effect, showing how the combination of genotype and treatment deviates from what one could expect based on their individual effect. So that brings me to the end of this video. I hope I was able to adequately explain the main effects, the interaction terms in design, parameter of the DESeq2 analysis, and hopefully I was able to explain when to use interaction terms, when it is best to not use them. And I hope it helps you to set up your design more appropriately depending on the biological question that you are asking. I've also created a small R script with small snippets of reproducible code demonstrating the cases that I went through in this presentation, which I will be uploading to my GitHub repository, and I will link my code in the description section below. So if you've enjoyed this video, please consider giving it a thumbs up. Don't forget to subscribe to my channel for more bioinformatics related content. If you have any questions or suggestions, please consider leaving it in the comment section below. Share this video with your friends and co-workers if you found this helpful. Please stay tuned for another exciting bioinformatics videos. I really appreciate your support, and I'll see you in the next video.